DNA is made up of nucleotides, arranged on top of each other like Legos. Nucleotide is made up of 3 parts: a 5-carbon sugar, a phosphate group connected with a covalent bond to the sugar's 5th carbon atom, and one of the 4 possible nitrogen-containing bases connected with the covalent bond to the sugar's 1st carbon atom. The nitrogen base could be either a single-ring pyrimidine, either a thymine or cytosine, or one of the double-ring purines, adenine or guanine. When the nucleotides are linked together in a strand of DNA, the phosphate group attached to the 5th carbon of the deoxyribose sugar of one nucleotide becomes joined to the 3rd carbon of the sugar of an adjacent molecule. The strand in which the sugar alternate with the phosphate is called the sugar-phosphate backbone. The backbone is not symmetrical. It has a definite orientation with a free 3' hydroxyl group at one end and a free 5' phosphate group at the other. The purine and pyrimidine bases stick out to one side of the sugar-phosphate backbone only. Each adenine molecule corresponds to a thymine linked together with 2 hydrogen bonds, while the guanine molecule corresponds to a cytosine base linked to 3 hydrogen bonds. That means that the number of adenine bases equals to the number of thymine, and the cytosine bases equals to that of guanine. The helical structure of DNA was actually first studied by Franklin using X-ray diffraction technology, and then later on developed by Watson and Crick. They actually imagined the DNA structure to be similar to that of a ladder, with the sugar-phosphate backbone as the sides and the nitrogenous bases as the rung. And since the purine, the larger molecule, corresponds to a pyrimidine on the steps, then all the DNA molecule has the same width and all of the nitrogenous bases are projected inwards. They also suspected that the anti-parallel nature of the DNA is responsible for its stability, with the 3' ends at opposite poles of the strand. Finally, the strand is wound, forming turns, with each turn containing 10 nucleotides.